Hi, I'm Dave Piston, the chair of cell biology and physiology at Washington University in St. Louis. Today, I have the distinct honor of introducing Dave Pagliarini, the 2021 recipient of the Earl and Thressa Stadtman Young Scholar Award. To me, this award is very fitting for Dave, since his approach to biochemistry research and training is a great example of the Stadtman way. If you aren't familiar with the Stadtman way, you might want to check it out on the NIH website in the history.nih section. Dave did his undergraduate degree at University of Notre Dame with a major in biochemistry, and then wound his way through graduate school, first at the University of Michigan and finishing at UC San Diego, doing his thesis research under the guidance of Jack Dixon. During this time, he discovered a novel mitochondrial phosphatase called PTP-MT1 and showed that it played an important role in ATP production and insulin secretion from the pancreatic beta cells. Having caught the mitochondria bug, he chose to do his postdoctoral research with Vamsi Mutha at Harvard. Dave set a goal at this time to find mitochondrial proteins with unknown functions that could play a role in mitochondrial diseases. Towards accomplishing this goal, Dave combined computational biology with rigorous biochemistry and created an annotated compendium of mitochondrial proteins, which he called MitoCarta. Launching from this data set, Dave worked to understand the biochemical mechanisms and how they affect cellular biology and physiology. In his own lab, he's continued this work and focused largely on the biosynthesis and transport of coenzyme Q. Through this work, the Pagliarini lab has discovered the functions of multiple orphan proteins. Importantly, each of these proteins has been shown to play a role also in mitochondrial disease. Here at Washington University in St. Louis, the essence of his lab continues to be systems biochemistry, really a blending of large-scale discovery science with mechanistic biochemistry. The lab is also moving into translational research, exploring potential small molecule therapeutics based on the discoveries from the lab. I'd like to call your attention to a actually wonderful biography that was written by Galara Venuzuela on the website. And it's available on the website for the program. And without further delay, here's Dave Pagliarini.
Okay, well, I'd like to begin by thanking Dave for that introduction and for recruiting me to Washington University recently. I'd also like to thank Brad Schwartz, who is the director of the Morgridge Institute in Madison, Wisconsin, and who originally nominated me for this honor. I'd also like to thank my anonymous nominators and letter writers. I'd of course like to thank the members of my laboratory, a tenacious and energetic group who perform the work I'll discuss with their own hands and who make the process of discovery so rewarding and so much fun. Now, I don't have time to tell you about all the different things we do in my laboratory today, which spans large-scale biology to cellular and organismal physiology to mechanistic and structural biochemistry. Today, though, I'd like to focus on this one theme, our systems biochemistry approaches, how we dive into the unexplored areas of mitochondrial biology and try to determine what orphan proteins do. And a broader theme that I hope to convey to anyone listening to this talk is that in science, there is a lot of unexplored space. And I'd encourage anyone, especially the earlier stage scientists listening to this talk, to venture out and to seek your own fertile land to explore. Now, to grasp the extent of this unexplored space, one has to look no further than the human genome. Back when I was a graduate student, the human genome was first published. And at the time, there was a sense that now with the genome being complete, now that we have this catalog of all these genes, that soon we'd understand the function of all of them. But 20 years later, the message is a bit different. The message is that we still know so little about them. And why is that? Well, in short, the answer is it's really hard to go in and identify the function of an unknown gene. There's many reasons for this, but there's lots of unexplored space. And my first exposure to this was back as a graduate student in Jack Dixon's laboratory. Now, if you don't know Jack, I would be remiss not to point out that he is the quintessential ASBMB scientist. He is the past president in '96. When I was in his laboratory, he won the William C. Rose Award. Soon thereafter, the Merck Award, and more recently, the Earl and Thressa Stadtman Distinguished Scientist Award, which really is the sister award to this one. Now, a little before I joined the lab, Jack published this little JBC paper. And the discovery here was that this phosphatase, this tumor suppressor phosphatase called P10, was not, as expected, a protein phosphatase, but instead was dephosphorylating a lipid second messenger, phosphatidylinositol triphosphate. Now, this JBC paper has been cited a mere 3,600 times as of this morning. And my thesis project became to look for other ones that are like it, to take advantage of this newly sequenced human genome, to dive in and to try to identify other phosphatases that might have P10-like qualities. I found one and was able to clone it and define some of its biochemical activity and get some clues to what it might be doing, but I still really couldn't peg down what this phosphatase was all about. And I then did a rather simple experiment, which at the time not only changed the course of my project, but really, in retrospect, changed the course of my scientific career. I simply asked, where does this phosphatase reside in cells? And the answer I got was this. And I didn't know what it meant. I had no idea what I was looking at, but it's now quite clear to anyone in the field that these are mitochondria. And then through my own work and through the work of a great postdoc in Jack's lab, Ji Zhang, it became clear eventually that, yes, this is another lipid phosphatase, that this PTP-MT1, as you call it, performs a key step in the biosynthesis of cardiolipin, which is a quintessential mitochondrial lipid that resides typically at these areas of high curvature in the cristae, a very important lipid. And here is this pathway that was known for a long time, but had this key missing step. Now, this discovery set off a whole new line of inquiry for me, because as a naive graduate student, I thought that there were no unexplored proteins in mitochondria. So I became very interested in the question of, well, what other proteins are out there? What else don't we know about this classic organelle? And so to begin answering that, I went to Vamsi Mutha's laboratory at Harvard Systems Biology and the Broad Institute. Now, at the time when I arrived, there were about 600 proteins annotated as being mitochondrial in the human genome. But we had good reasons to believe there were more. And so we began this systematic approach that first started off with deep proteomic analysis of purified mitochondria from different tissues that we then integrated using computational approaches with other clues that we could get throughout the genome. Then with microscopy and literature searches, and basically by the end came up with what we called our mitocarta compendium of mitochondrial proteins. And it's, although imperfect at the time, it almost doubled the number of known proteins in mitochondria. And immediately we could put this compendium to good use because we knew there were gaps in knowledge about mitochondrial function. For example, complex 1 is the first complex in the respiratory chain. And it is inborn errors of complex 1 are among the most common inborn errors of metabolism worldwide. Now, to make complex 1, you have to assemble it from these different parts. And we knew that there are all of these proteins out there that are responsible for this assembly process that were unidentified. That is, that patients that have complex 1 deficiencies often don't have problems in complex 1 itself, but in these assembly factors. And we're able to identify a new one that we called assembly factor 5 and show that mutations in this gene underlie a human condition. Now, there are many more uncharacterized proteins. We call these MXPs, mitochondrial uncharacterized proteins. At least a quarter, conservatively, of the mitochondrial compendium was uncharacterized. And then as we explored further, we could find all kinds of pathways similar to the cardiolipin story, where there were missing steps. We knew the pathway existed, but we didn't know all the steps that were involved. This here is a cartoon of the coenzyme Q biosynthetic pathway. Now, importantly, a third fact is that all of these patients, as I mentioned, there are so many whose condition lacks a known causal variant. So we don't know why these patients are getting sick. And there are no FDA-approved therapeutics for mitochondrial diseases. So these four facts really form the foundation of my laboratory and what we've been doing for the last 11 or so years. That is, we begin with these MXPs. We begin with our understanding of the mitochondrial framework and the missing steps therein. And through some sort of systematic functional characterization, we try to associate these MXPs with known pathways and processes. Now, there's no one way to do this. There's no one magic formula. So we keep changing our approach, keep changing our upfront screening techniques, a couple of which I'll tell you about today. Now, importantly, after we make a connection, we don't want to stop there, but we want to then really dig into the mechanistic and structural biochemistry. We want to work with clinical geneticists to see if these proteins can explain, mutations in these proteins can explain some of these unresolved diseases. And eventually, the goal, of course, is to use all this new knowledge to come up with some new therapeutic approaches that are badly needed for the field of mitochondrial medicine. So I'll tell you about a couple of these today, but I'll focus on a subset of mitochondria here. This is a brief, high-level overview of cellular metabolism, glucose being converted to pyruvate through glycolysis, pyruvate feeding the TCA cycle to create NADH and FADH2 for the electron transport chain. Eventually, the reduction of oxygen to water at complex 4, and the coupling of reentry of protons to the production of ATP at complex 5. Now, I'll just focus today on two simple parts, complex 1 and coenzyme Q. Plenty of gaps in knowledge in both of these fields and many unresolved diseases involving their function. So how do we begin to explore this? How do we dive into these unexplored proteins and see if they could explain some of the missing biology? Well, here's one example. The first I'll tell you about is infinity enrichment mass spectrometry. So one of the first things we've done is to take a collection of these MXPs, 50 to be exact, and some characterized controls, and systematically try to purify them and see what comes along in 2 different cell lines under 2 different nutrient conditions. Now, I'm certain that anyone listening to this talk who has studied a protein has done an experiment, something like this along the way. And frustratingly, what typically happens if you take your favorite protein, purify it, hoping to get some magic hit, you don't get 1 or 2 things, but like us, you would get something like 1,400 different hits. Now, this doesn't exactly lead you to the next steps, but if you do these enough times and you do enough of them, here 1,000 different experiments, you could very efficiently begin to prioritize which of these dots are real bona fide important interactors and which ones are kind of spurious. You could do this because you could assess the specificity, reproducibility, and the abundance of these interactions. So for us, by the end, after getting through 1,000 of these, we could effectively prioritize these 1,400 down to about 10. And the top interactor was this assembly factor that I just told you we associated with the human disease after the myocarditis study. So here, this leads to a very specific hypothesis that this protein is another assembly factor for complex 1. Now, we can test this. In the field, we do an analysis that perhaps strangely is called a Seahorse analysis, where we could take live cells and measure the oxygen consumption rate. Here are live cells. We're measuring their rate of oxygen consumption. We can uncouple them, which is tantamount to putting your car in neutral and revving the engine to see how high you can get the RPMs. This is maximal respiratory capacity. We can then poison them with a complex 1 inhibitor that shows how much of the oxygen consumption is due to complex 1. So these are wild type cells. Now, if we silence our new protein, we see something very different. Low baseline respiration, no spare respiratory capacity, very little residual complex 1 because these aren't full knockouts. And we can also see why. We've renamed this protein as assembly factor 8. And when you lose this, you lose its binding partner. So once we got to this point, now we're confident that this is really something that's very important for complex 1. Now we could talk to our clinical genetics collaborators and say, have you ever seen this protein? Do you have any suspicious variants in this gene that might underlie human condition? And they said, well, this is a small gene, 3 exons. In one of our cases, there's clearly a disruption, a mutation in the maternal chromosome that throws this out of frame. This would, of course, be disruptive, but these are recessive disorders, and the paternal chromosome looks fine. But given our new data, they resequenced the entire gene and found a deep intronic variant that somehow leads to complete loss of expression from this allele. So now we're able to get cells from the patient and see that the condition here is really complex 1 specific. The rest of the complexes seem generally fine. And now we could do the key experiment by putting back into the patient cells this a wild type version of the protein we've identified. When we do that, we rescue complex 1 activity and we rescue the protein with which it interacts, essentially proving the pathogenicity of the mutation that we've now found in various cohorts worldwide. So that worked there. This was an example of affinity enrichment mass spectrometry really helping us to deorphanize one of these proteins. But that doesn't always work, and so we have to move to other methodologies. Another one I'd like to tell you about is this one we call multi-omic profiling. So again, sort of the challenge here is the same, starting with our uncharacterized mitochondrial proteins, represented by dots. So here are 50 of them. Here are many more characterized genes. And now we've taken this to yeast. And in yeast, we're making knockout of one of these genes in each yeast strain, growing the yeast under 2 different nutrient conditions, and profiling these as deeply as we can using mass spectrometry-based methodology in collaboration with our collaborators, the Kuhn Lab in Madison. We call this the Yeast 3000 or Y3K study because of the 3 times 1,000 measurements here. And of course, this gives you lots of data. Here I'm showing along the top, the columns are all different strains. The rows are all different biomolecules that we can measure. And there are all kinds of sophisticated analyses that we or others can do to learn new biology from this data. But I'd like to point out 3 really straightforward, simple approaches that have worked very well for us. And the first is just to start with a molecule of interest. If you have a molecule or a protein, lipid, metabolite, whatever, that you're very interested in, you could ask, are there any of these strains that we're studying that have a special relationship to that molecule? Essentially, are there any strains, each represented by a dot here, where this molecule behaves differently, an outlier? The second thing you can do is ask, how do pairs of molecules behave coordinately or anti-coordinately across the whole data set and come up with these correlation maps? And the third is to ask to compare full strains and say, well, here's a strain whose gene disruption is we've disrupted a gene of unknown function, but here's one with a known function. Are they correlated? It turns out that when genes are knocked out within similar pathways, they have a very highly correlated whole cellular response, such that if you did not know that gene B were related to gene A, you can infer it from this data. So I'll give you one example of this outlier. So again, here, we're looking at the changes in one molecule across all knockouts. And the molecule we're looking at is something called 4-hydroxybenzoate. It's a precursor to coenzyme Q. That for more than 40 years, we have not understood how cells take tyrosine and turn them into this precursor. Now in our study, only once whether was there a knockout strain where this molecule was decreased significantly in abundance. And it led to a very clear hypothesis that this enzyme acts at this specific step. Now we could test this. We could test this genetically by saying, can we rescue cells deficient in this enzyme with this molecule or with anything upstream? And it's really only this molecule that rescues the growth of these cells. And biochemically, we can purify it and show it does all the requisite biochemistry to explain this. And then through other experiments really have proven that this is the missing step in this pathway. Now, through the other analyses and other similar analyses, we're taking this whole pathway from tyrosine all the way down to coenzyme Q and learning all the different proteins that are involved in this pathway and those that regulate this pathway. But I began by telling you that a larger problem are that so many patients with mitochondrial disorders don't have a molecular diagnosis. Even when we do exome sequencing, we only get a successful diagnosis in about 30 to 60% of patients. So in more recent times, we have been asking, can we take this technology to human cells? Now, in unpublished data, I'll tell you about how we've been doing that. The approach is similar. Now here, we're starting with a human cell line, and now each of these dots represent gene disruption. We call this our mitochondrial orphan protein multiomic Crispr screen, or mitomics study. So Crispr, of course, has been used with high success for many forward genetics applications, but this represents a reverse genetics approach, where we've individually knocked out these genes. Again, we have uncharacterized mitochondrial proteins and what we call our sentinels, those that are whose function we understand pretty well. And we again profile these lines using the best mass spectrometry techniques we can. And really, in each cell line now, we can measure more than 12,000 different molecules. So very dense data for every one of these cell lines. So again, how do we use this? So many different ways we could use this data, but starting with this problem of that we know there are patients with complex 1 and coenzyme Q related disorders without causal variants, we could ask, are there any of these proteins that we can connect to complex 1 or coenzyme Q? And a way we could do this is essentially to go fishing in the data where our bait is the molecule of interest. For example, coenzyme Q. Do any of these knockouts result in coenzyme Q deficiency? Well, we ranked those genes, and the gene with the largest decrease in coenzyme Q that was not already known to be involved in coenzyme Q was something called PURF, a really terrible name that doesn't mean much. Now, remarkably, when we asked the same question about complex 1 proteins, we got the same answer. PURF was our top hit. Now, because we've done all the proteomics, we could ask what is happening on the proteomic level here. So here is a volcano plot, as we call it, just looking at fold change for the loss of this protein, which PURF, we used to call it MXP17, and a measure of significance. Now, the top hits are all things involved in coenzyme Q and complex 1 deficiency. And if you're really paying attention so far, you would note that Endof AF5 was the first protein I told you about. C17 ORF89 was the second protein I told you about. So they're already related. And now we're relating this to a third protein. Now we could also begin to layer in our former protein-protein interaction data and say, have we ever seen this protein interacting with anything? Well, across more than 100,000 different binary interactions, this protein, PURF, MXP17, only showed up twice, like 2 times. The first time it was found to interact with Endof AF5, and the second with CoQ5, and maybe a bit with CoQ3. Now, each of these proteins then react with their respective networks. Now, what's remarkable is that Endof AF5, CoQ5, and CoQ3 are all the same kind of protein. They are all SAM-dependent methyltransferases. So now here's this protein, this small protein, that somehow is affecting the methyltransferases in both of these pathways. So we again called up our collaborators, Rob Taylor, his former student, Charlotte Alston, and said, we have another case here. Have you ever come across this gene? And soon thereafter, identified a patient family with a variant here. This patient, unfortunately, had passed away, so we could not get patient material, but instead we were able to CRISPR engineer this mutation into these cells and recapitulate the same phenotype, the loss of coenzyme Q proteins, the loss of complex 1 related proteins. So essentially what we're finding here is that these two pathways, this assembly of complex 1 and the mitochondrial production of coenzyme Q, both of which rely on methyltransferases and which obviously work very closely together. Complex 1 passes its electrons to coenzyme Q. These pathways, these highly related pathways, are regulated by a common protein called PIRF, which we've now renamed Endof assembly factor Q. So across a series of these studies, we're beginning to understand how complex 1, all 45 subunits are shown, are assembled module by module by these different assembly factors. I showed you that one technique, phylogenetic profiling, as it turned out, helped us get the first one. A second, affinity enrichment mass spectrometry, helped us get another one. And now this multi-omic profiling is helping us get a third. There are others, and of course, we continue to uncover more of these with their data. Now, there are lots of other things I could tell you about this data. I don't have much more time to tell you, but one thing comes full circle back to our phosphatases. PTP-MT1 is one such phosphatase in the mitochondria. But it turns out that with mitochondria, we've discovered multiple others. There are multiple other phosphatases that are resident. They live either inside or in the intermembrane space, perhaps in the outer membrane. They are part of different phosphatase families, which have different preferred residues. And these are part of our mitomic studies, such as PPTC7, excuse me. And amazingly, there are now 8,000 some different mitochondrial phosphoisoforms, but only a very small fraction do we have any semblance of their function. Most of these, even these, have very little understanding what they actually do. Now, recently, we made a knockout of one of these phosphatases. The mice are born in medallion ratios, but within 1 day, they are all dead. They have severe disruptions to their mitochondrial content. They're small, they lose half their content. They're major targets in import machinery, fatty acid oxidation, the TCA cycle. And I'll just point to an upcoming JBC mini review that we have written about this. Now, with that, I'd like to thank a lot of different people. This is a very COVID era acknowledgement slide where we can't all be together, but I have just an amazing laboratory, amazing collaborators that include Josh Kuhn's lab, and Rob Taylor, our clinical geneticist collaborators, alumni that have led some of the work that I told you about, my former advisors, again, Dave and Brad, for their introductions and nominations, my funding, ASBMB, thank you for being such an amazing organization for promoting and advancing our field. And of course, we are always looking for new talent to join our initiatives. Thank you.